morning everyone. Today we are going to be discussing about the oxidative phosphorylation and electron transport chain. Oxidative phosphorylation is defined as the formation of ATP from ADP and inorganic phosphate coupled with electron transport chain. Now we have to be discussing about the fundamental information regarding oxidative phosphorylation. That is, we have to be learn in detail about the bioenergetics. What is bioenergetics? Bioenergetics are biochemical thermodynamics which deals with the study of energy changes in biochemical reactions. The reactions are broadly classified into two. One is exergonic reaction, that is energy releasing, and endergonic reaction, that is called as energy consuming. So by the end of these kind of reactions, what is resulted or what is formed is energy will be resulted. That is, we have to be discuss about how the high energy compounds are formed. Certain compounds are encountered in the biological system which on hydrolysis yield energy. The term high energy compounds or energy rich compounds is usually applied to substances which possesses sufficient free energy to liberate at least 7 calorie per mole at pH 7. So we have to be discuss about the ATP and the ADP cycle, which based on the oxidative phosphorylation and substrate level phosphorylation. We already been discussed about the oxidative phosphorylation, and during this oxidative phosphorylation, ATP generation is relatively higher than the substrate level phosphorylation. That means substrate level phosphorylation is otherwise called as the uncoupled oxidative phosphorylation, so that the energy will be comparatively lesser than the oxidative phosphorylation. The ATP formed or high energy phosphate formed during these reactions is utilized for the different biochemical functions, that is muscle contraction, active transport, biosynthesis, and phosphorylation reactions. The hydrolysis of ATP, which is associated with the release of large amount of energy, that is, when ATP on hydrolysis, it is converted to ADP and inorganic phosphate. As a result, 7.3 calorie of energy will be liberated. The liberated energy is utilized for the various processes like muscle contraction, active transport, etc. ATP can also act as a donor of high energy phosphate to low energy compounds to make them energy rich. On the other hand, ADP can accept high energy from the compounds possessing higher free energy content to form ATP. So that ATP, which acts as an energy link between catabolism, that means degradation of molecules, and anabolism, that means synthesis of biological system. So how this ATP will be synthesized? Already we discussed about the two processes. One is related to oxidative phosphorylation, another is by substrate level phosphorylation. That means during oxidative phosphorylation, the ATP generation is higher, and substrate level phosphorylation comparatively the ATP generation is less. So we have to find out the examples for high energy phosphate compounds. Examples are phosphoenol pyruvate and 1,3-bisphosphoglycerate. These are the compounds, or these are the intermediates which are formed during glycolysis. And to find out the succinyl-CoA is the substrate level phosphorylation compound intermediate in the TCA cycle, which can transfer high energy phosphate to ultimately which produce ATP. So when you go through this flow diagram, we can find out carbohydrate, fats, and amino acids, which is ultimately converted to carbon dioxide into water during oxidation. So in these reactions, NAD get reduced to NADH and FAD get reduced to FADH2. As far as NAD is concerned, there is a free proton will be released. That free protons are transported to ETC and which get hydrolyzed to form ATP molecule. So as far as the electron transport mechanism is concerned, it has 5 distinct complexes. The first complex which is related to NADH coenzyme Q, and second complex is related to succinate coenzyme Q, and complex 3 is associated with the coenzyme Q cytochrome C, and complex 4 is concerned with the cytochrome, and complex 5 is related to ATP synthesis. So in these complexes, which is also been related to different enzymes. One is NADH coenzyme Q reductase, second is succinate coenzyme Q reductase, and third is cytochrome coenzyme Q reductase, and fourth is cytochrome oxidase, and fifth is ATP synthase. So these reactions in which substrate get oxidized, which the electrons are transported to NADH through the NADH, it is reaching the complex, there it binds with the iron sulfur compounds, and these electrons are centralized in the coenzyme Q. So same thing may be happened in the succinate compound also, that is complex 2, which is related to FADH2 and the iron sulfur complex. This is pooled in coenzyme Q, and is due to the high concentration of electrons in the coenzyme Q, it is little transported from the complex 1 and 2 to complex 3. There it is transported via the cytochrome B, iron sulfur complex, and cytochrome C1 by the help of the enzyme cytochrome coenzyme Q reductase. Then the electrons are transported to coenzyme C, thus it is reaching the complex 4, that is cytochrome oxidase. There it gets oxidized to H2O, that is what is produced as a result of combination of hydrogen and free oxygen. So the free electrons which is produced from the complex 1, 2 and 3, which is pooled in the complex 5, there ATP synthase enzyme is there through the F0 and F1 particle of mitochondria. There ADP combines with inorganic phosphate which forms ATP. Thus it is ATP is generated. Thus generated ATP is stored in the mitochondria, so that mitochondria is considered as the powerhouse of the cell. So these are the complex in which through these complexes the electrons are normally being transported. As a result, substrate, from the substrate electrons are transported to NAD to FMN. There ATP is produced, that is what is called as a site 1 ATP synthesis, and FMN to coenzyme Q and cytochrome B, it is called as site 2 ATP synthesis, and cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3, there and cytochrome A3 ATP synthesis, that is called site 3 ATP. So there are 3 sites at which ATP is normally being synthesized in the electron transport system, that is FMN, cytochrome B, and cytochrome A3. So some of the inhibitors are there. These inhibitors which may completely inhibit or partially inhibit the transport of electrons from complex 1 to complex 2, that is FMN to cytochrome B, the amytal, pyridine, rotenone, all these are the classical examples for site 1 ATP inhibitors. And cytochrome B to cytochrome C1, amytal and British anti-leavisite, these are considered to be the inhibitors which will reduce or completely inhibit the transport of electron from cytochrome B to cytochrome C1. That another is the cytochrome A3, that is called as complex 3. From this cytochrome A3 to molecular oxygen, cyanide, carbon monoxide, and sodium acid, these are considered to be the powerful inhibitors which is completely inhibiting the flow of electrons from the cytochrome A3 to molecular oxygen. So this is called as the terminal inhibitors. This oxidation process is called as terminal oxidation. So the energy rich compounds which may be carbohydrate, fats and amino acid, which may undergo series of metabolic reactions which may finally get oxidized to carbon dioxide and water. The reduced equivalents from the various metabolic intermediates are transferred to coenzyme to NAD and FAD to produce respective NADH and FADH2. That means it gets reduced. The latter two reduced coenzymes passes through the electron transport chain or respiratory chain and finally reduced oxygen to water. The passage of electrons through ETC or electron transport chain is associated with the loss of free energy. A part of this free energy is utilized to generate ATP from ADP and inorganic phosphate. When you see the structure of mitochondria, as you are aware about the mitochondrial compounds which present or components which present, which has the different distinct parts. One is the outer membrane, inner membrane. The inner membrane has a finger-like projections inside, that is called as cristae, and central part is called as matrix. So in the inner mitochondrial membrane, you can see NAD, FMN, coenzyme Q, cytochrome B, cytochrome C, cytochrome A, and cytochrome A3. These are assembled in a sequence. This is what we call as electron transport assembly. Okay, through these electron transports, normally electrons are transported and it is reaching the F1 particles here. There ATP synthesis there get phosphorylated, thus it is forming ATP. Thus formed ATP is stored in the mitochondria. So the mitochondria is considered to be center of metabolic oxidative reactions to generate reduced coenzymes, that is NAD and FADH2, which in turn are utilized in ETC to liberate energy in the form of ATP. For this reason, mitochondria is appropriately regarded as the powerhouse of the cell. When you see this, we have several concepts or theories there to explain the electron transport mechanism, and some of the compounds or some of the concept or what we call as the hypothesis are being generated to well explain this concept or this mechanism. One is what we call as the chemi-osmotic hypothesis. This is the most accepted hypothesis in which we can see this mitochondria, that is mitochondrial membrane, inner mitochondrial space, inner mitochondrial membrane, and mitochondrial matrix. So if you see the inner mitochondrial membrane, in which the complexes 1, 2, 3, 4 complexes are there. Of these, complex 1, 3 and 4, which is related to the release of free H plus ions in the outer membrane, 2 plus 2 plus 2, 6 hydrogens pooled together, and when the concentration of hydrogen is increased in the outer membrane, it will diffuse or penetrate to the inner membrane. There it is phosphorylated, thus it is forming ATP. That is the one of the concept is well established or well accepted to explain this concept. The mitochondria normally consists of 5 distinct parts. These are the outer membrane, the inner membrane, and inner mitochondrial space, and cristae and the matrix. So in this the cristae, there you can find a small inner finger-like projections. These are there you can find the abundant amount of ATP synthase enzyme. That ATP synthase enzyme is responsible for the synthesis of ATP. Thank you.